Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about interrupted genes. Most of the eukaryotic genes contain non-coding sequences known as introns that interrupt the coding sequences or exons. Because of this, such type of eukaryotic genes are known as interrupted genes or split genes. These non-coding sequences are excised from the primary transcript prior to their transport to the cytoplasm. Most of the well-characterized prokaryotic genes contain a continuous sequence of nucleotide pairs that specify a collinear sequence of amino acid in the polypeptide chain. However, in 1977, the molecular analysis of three eukaryotic genes showed the presence of introns. These three eukaryotic genes are mouse beta-globin gene, rabbit beta-globin gene, and chicken ovalbumin gene. In 1977, the molecular analysis of these three eukaryotic genes indicated the presence of non-coding sequences or introns. Subsequently, it was found that these introns are present in the non-translated regions of the gene. Some of the earliest evidences regarding the presence of introns came from the electron microscopy of DNA-RNA hybrid. Friends, RNA-DNA duplexes are more stable than the DNA double helix. So when a double-stranded DNA is partially denatured and the partially denatured DNA is incubated with homologous RNA molecules, hybridization occurs between the homologous RNA and the complementary DNA sequence. So here friends, hybridization has taken place between this homologous RNA and the complementary DNA strand that result in the formation of DNA-RNA hybrid. This DNA-RNA hybrid contain a single-stranded region. This is the single-stranded region which is known as R loop. This R loop is formed by the displaced strand. During the formation of this RNA-DNA hybrid, the equivalent strand has been displaced, and this displaced strand has formed the R loop. So friends, in this figure, this is the double-stranded DNA. This double-stranded DNA is partially denatured. After that, the partially denatured double-stranded DNA is incubated with homologous RNA. After that, hybridization occurs. So the DNA-RNA hybrid is formed, and this is the R loop. Shirley, Tillman, Philip Leader and colleagues, they conducted a number of hybridization experiments. In the first experiment, they incubated mouse beta-globin gene with beta-globin mRNA. This red colored strand is the mouse beta-globin mRNA, and this blue color molecule is the mouse beta-globin gene. So after incubating these two under suitable condition, hybridization occurred, and they found the presence of two R loops that are separated by a loop of double-stranded DNA. This is the loop of double-stranded DNA, and these are the two R loops, and this red color strand is the beta-globin mRNA. So the presence of this loop of double-stranded DNA indicated the presence of a non-coding sequence in the middle of this beta-globin gene that do not encode amino acid in the polypeptide chain. These are the two loops, and this is a loop of double-stranded DNA. This loop of double-stranded DNA contains the non-coding sequence. So hybridization do not take place between this non-coding sequence and the beta globin mRNA because this beta globin mRNA do not contain introns. These introns have been already excised during processing. Similarly, they conducted another hybridization experiment. They incubated mouse beta globin gene and beta globin pre-mRNA under suitable condition. So here also hybridization occurred. And the hybridization between the mouse beta globin gene and beta globin pre-mRNA resulted in the formation of a single loop of single-stranded DNA. The formation of this single-stranded DNA loop indicated that the beta globin pre-mRNA contain both coding sequence and non-coding sequence. Because the beta globin pre-mRNA contain both coding sequence and non-coding sequence, so only one loop of single-stranded DNA was found. By comparing these two experiments, Tillman and co-workers, they concluded that these non-coding sequences, which are known as introns, are removed by the processing events that convert pre-mRNA to a mature mRNA. Later on, Tillman and co-workers they compared the nucleotide sequence of beta globin gene with the predicted amino acid sequence of the beta globin polypeptide chain. On the basis of this comparison, they suggested that there is a single intron in the beta globin gene. But subsequent research indicated that this beta globin gene contained two introns. So friends, this is about the discovery of introns. Now we shall know about the biological significance of introns. At present, scientists know little about the biological significance of introns. However, introns may be involved in the regulation of gene expression. Similarly, introns provide and selective advantages by increasing the rate at which the reassortment of coding sequences can take place by recombination, thereby speeding up the process of evolution. So friends, these are few biological significance of introns. For more updates, please subscribe SAM online class. Thank you very much friends. See you again. Good night. Bye bye.